Hi, fellow student and professor. I'm Zheng Xuan from National Tsinghua University Dr. Yu-Chun Ling's lab. I'm very excited to share here with you our research about branch actin filament form size-dependent diffusion barrier around centrosomes. In this study, we ask, as microtubule organizing center, platform for ciliary formation, and various cellular signaling, how do centrosomes maintain their microenvironment without membrane in the crowded cytoplasm? In the following experiment, we investigated centrosome at three subcompartments, from core to the exterior region, central lumen, pericentral matrix, and the central satellites. On top of it, we incorporated chemically inducible dimerization system, in which FRB and FKBP can join together by rapamycin. Such dimerization allows us to bring protein of interest to designating locales as we conjugate them with the parts. We associated FRB with centrosome marker and FKBP with probes. Theoretically, without diffusion barrier, rapamycin can trap diffusive probe at centrosome. Subsequently, we will see accumulation of probe on targeted area. On the other hand, presence of diffusion barrier can hinder the access of diffusive probes, and therefore we see no accumulation. Ideally, in the following experiment, the centrosome targeting protein should not participate in the diffusive process or cause active transport. We select considerably stable centrosome targeting protein using FRAP assay. Subsequently, we use centrin 2 located central lumen, CDK5-Rep4 PAC domain, PAC in short, at pericentral matrix, and PCM1F2 at central satellites in the following experiment. We recruited probes ranging from stoke radius 3.2 to 7.6 nanometer to each site. I will abbreviate these probes by their size, calling them the probes 3.2 or probe 4.1. First off, all probes to central satellites, which are marked in blue. Probes were tagged with YFP from small to large. Probes rapidly translocated within half a minute post-rapamycin addition. And then all probes to pericentral matrix, which are marked in blue. Interestingly, the larger the probes, the slower for the accumulation to reach plateau. It's now already less likely to see accumulation of two bulky probes, indicating obstructed diffusion. Similarly, all probes to central lumens, tagged by centrin 2 in blue. Consistently, bulky probes show less or no translocation. We argue that 3-minute post-rapamycin treatment was perhaps not enough for visible accumulation for bulky probes, so we extended the time and recorded translocation 2 hours post-rapamycin treatment. Coherently, probability of probes translocation noticeably dropped for large probe at the same locales. Therefore, we inferred that the barrier lies outside pericentral matrix. We then tried to investigate the composition of the barrier. We postulated that microtubule and actin, the two rich elements around centrosomes, may be the major components. Hypothetically, removal of the major component would significantly change the permeability pattern. To start, we used nocodazole treatment to disrupt microtubules. The diffusion pattern remained unchanged. Following, we disrupted actin with CK666 and latrunculin A respectively to examine actin's role. Actin filaments are marked in red, centrosomes are in blue. Image and normalized mean intensity of F-actin around centrosomes suggested that our CK666 and latrunculin A treatment significantly disrupt actin structure. From left to right are each centrosome subcompartment from exterior to core, marked in blue. Green fluorescence is probe 6.3. The image demonstrates that as the bulky probe cannot diffuse across the barrier, diffusion was observed after actin disrupting by CK666 and latrunculin A. The quantitative data shows that actin disruption had significantly increased the translocation probability for bulky probe 6.3 and 7.6. The change in the diffusion pattern after disrupting the actin filament had us identify and conclude that actin filament forms centrosome diffusion barrier. We further ask whether it works the opposite way. Does rising actin level around centrosome decrease barrier permeability? Therefore, we took advantage of the temporary increase of actin filament during early anaphase and synchronized ourselves for the following experiment. Usually, probe 4.5 is small enough to diffuse across the barrier. Therefore, we collected interface and anaphase cells. Pericentral matrix is marked in blue, probe 4.5 in green, and chromatin in red. The videos demonstrate that diffusion was obstructed during anaphase. Quantitative data showed that the diffusion of both probe 4.5 and 6.3 is significantly hindered. The results suggest that increase in actin around centrosomes during anaphase has temporarily decreased permeability of diffusion barriers. On top of this result, as suggested by image and quantitative data here, we use CK666 to perturb the anaphase actin filament and had the barrier permeability increase again to allow access to the same probe. We have come to infer that permeability of centrosome diffusion barrier is tightly regulated by branch actin filament around centrosomes. And now we wonder whether the permeability of such barrier affect microtubule and microtubule organizing center. Here we mark centrosome red and microtubule nuclear component the gamma tubulin green. Image and quantitative data indicate that disrupting diffusion barrier significantly increased gamma tubulin level at centrosomes. Although gamma tubulin was rather small in size, the observation encourages us to run further examination using GCP4 protein, a larger component in gamma TERT. We carefully evaluate the validity of using GCP4 to represent centrosome and gamma TERT in our following experiment. GCP4 delta C is the truncated form. Images and quantitative data show that compared to the truncated form, GCP4 did highly colocalize with centrosomes. We examined GCP4 dynamics using FRAP assay. As the image show, disrupting diffusion barrier increased GCP4 shutting rate at centrosome. Quantitative data suggests that the increase in mobile fraction was significant after disruption. The increase in microtubule nuclear consequently increased the number of microtubule emission from centrosome, while not affecting the elongation speed. In conclusion, our study demonstrated that there are size-dependent centrosome diffusion barrier lies between pericentral matrix and central satellites. The permeability of the barrier is tightly regulated by branch actin filament around centrosomes. The barrier gate microtubule nucleation by interfering with the gamma TERT recruitment. And that's all for my presentation. I would like to share more details with you during the poster session. Please join me, and thank you for your listening.